Chapter 19, Section 4, the mitochondria in thermogenesis, steroid synthesis, and apoptosis. We see that the uncoupled mitochondria in brown adipose tissue produces heat. Okay, brown adipose tissue. This is, the tissue in newborn mammals that functions to generate heat. Okay, this is what helps new mammals keep some, internal heat going. Because they don't have enough layers of fat. They don't have the glycogen in their, liver and their muscles yet. They don't have a lot of things there that we would burn, and in the process lose a lot of that to that second law of thermodynamics to keep our internal temperatures going. So this adipose tissue is what's going to generate a lot of this heat. The uncoupling protein 1, UCP1, it's a long chain fatty acid, hydrogen symporter. Provides a path for protons into the matrix, which will then, which aren't going to use the F0, F1 subunit complex that we know as ATP synthase. Phosphocreatine is also going to be used as for heat. So this creatine, phosphocreatine, as you're going through, cycling through, this is an extremely inefficient mechanism. Inefficient, which means second law of thermodynamics be damned. Boom. Lots of excess energy is going to be lost exothermically. Maintain an internal body temperature. Same thing we see here, uncoupling. So the flow of the hydrogens from out to in bypasses ATP synthase and goes through this. Things can then pump through, flow through. We get an extremely inefficient mechanism, which, again, second law of thermodynamics being damned, high loss. High loss exergonically leads to high loss exothermically, which means this animal will now have a lot of heat to keep itself generating heat. Hibernating animals will utilize these mechanisms. Burning of, from this adipose tissue, the brown adipose tissues. So the mitochondria, via this uncoupling, this different mechanisms of flow of hydrogens, flow of the electrons, as a means of wasting energy. Thermodynamically wasting, which means they will always be able to maintain certain level core internal temperatures, which so they don't freeze through. They're not taking in organic molecules like you do on a daily basis, and the breakdown of that and all those little bitty reactions, chemical reactions, losing little bits of energy due to the second law of thermodynamics means you, as you watch this, are maintaining 98.6 degrees Fahrenheit, 37 degrees Celsius. They're hibernating. They're not eating. What they are doing is they are burning adipose tissue. They are burning up those stored lipids. Well, in some of those adipocyte cells, they've uncoupled the mitochondria, which means that the mitochondria aren't there for making of ATP. The mitochondria are there for generating an internal temperature. We also see that the mitochondria is going to be central in steroid hydroxylation. So certain steroid precursors are going to be made here in the mitochondria. These biosynthetic reactions produce certain steroid hormones. You're going to see that these are the mitochondria in cells of the adrenal glands, the gonads, the liver, and the kidney. So no, this isn't the mitochondria of your muscles. This isn't the mitochondria of your skin epithelial cells. These are going to be certain cells in your adrenal glands, gonads, liver, kidney that are going to be producing certain steroid-based hormones, corticosteroids, testosterone, estrogen. Oh, hey, gonads, there you go. The cytochrome P450 family. These are a type of monooxygenases. The reactions, some R group plus oxygen, NADPH. You get a hydroxyl, water, NADP. Monooxygenases, single oxygen. Singlet O2s, one of the oxygens, what was hydrogen, now becomes a hydroxyl. That's what they are good for. That's what they do. Gonna have to bring in some helpers, a flavoprotein, some iron sulfur protein, and we start with NADPH, oxidized to NADP. FAD, protein, here adrenodoxin reductase, goes from has FAD with it. Adrenodoxin reductase now has FADH2 associated with it. And you go through a series of these. Reduce, FAD, FADH2, iron sulfur, oxidized, reduced, and then finally, ultimately, you can't see because of my big head, if you look at the handouts or the figure in the textbook, some R group that has a hydrogen will now end up with a hydroxyl. All of this change of hydrogen to hydroxyl due to this electron going through these stages to give us this right here. We see that hepatocytes, liver cells, have versions of these P450s in their ER, not their mitochondria. They catalyze similar reactions, similar, not the same reactions. They do some of the similar things. And we can also take this and we can do this xenobiotically. So it's not compounds, found in nature. These are going to be synthesized industrially. So we've modified enzymes. We've modified this, the pathway we just talked about, to do something similar. So we can make compounds that are not found in nature, but are laboratory derived. And that's how we get some of our current medicines, antibiotics, current compounds. Now, every one of your cells has within it the ability, the instructions on how to die, in a programmed way. Here's a certain series of steps that must be followed when you get the signal. And those signals are, death signals, is literally what they're called by immunologists. During your normal embryonic development, these kick in some external signal, like that death signal I just talked to you about, one of your immune cells, CD8 cytotoxic T cell tells one cell, hey, you need to die because you're virally infected. Well, it kicks in this apoptosis, this series of events of cell death. And it can be an internal signals, DNA damage, accumulation of those reaction oxygen species causes too much damage. Heat shock can induce this process. This process begins with a permeability issue. The membranes become so permeable that that proton motive force that we've just spent the entire chapter talking about dissipates because those hydrogens are lost literally to the cytoplasm. Depolarizes the mitochondria. Then you kick in with APAF1. These are protease activating factors. They complex with cytochrome C to form what is called an apoptosome. This apoptosome goes through and starts cutting and activating caspases. Caspases are a series of enzymes that go through, activate each other, and they have multiple steps. Each of these steps is another layer of complexity, which means more complexity, more controlling points, more regulation points. Each of the caspases, they activate, 9 activates 3, 3 activates 2 or 7, and depending on the cell, and then activates more and more and more. The numbers are just which ones they found in which order. 9 doesn't activate 8, 8, 7, all that, no. So you'll see, the caspases activate each other, but then not only do they activate each other, but then they go through and they also have, some of them can have steps. One acts as a protease, another acts as a nuclease. And they just basically start activating, they either do the damage themselves or activate the enzymes, which will then go and start cutting up the internal components of that cell. And all of this is kept in check because the mitochondria is healthy.